Okay, so I'm going to do a quick wrap-up podcast today on what we covered regarding the B cell and T cell receptors and kind of the beginning of their signaling pathway. So let's start with the B cell receptor. So here is the cell surface of the B cell. And get out all the colors here. So the B cell receptor is, of course, there are the heavy chains, right? And our light chains, right? And they're joined by disulfide bonds. And right, this tip is the variable region. And here's our constant region. Okay, and this is our B cell receptor, or BCR. Okay, and it's going to bind our antigen here. Here's our antigen. Okay. And this alone doesn't initiate signaling, so this antigen doesn't send a signal through the B cell receptor. It needs a co-receptor, and the co-receptor involves a variety of proteins. Okay, so I'll talk about each one, but let's put them all in the membrane. So we have Ig alpha and Ig beta. Okay, and I'm just going to make sure everything's in focus because we had a problem last week. So Ig alpha and Ig beta, and we have CD19. Pick a different color. Okay, and we have CD21. Okay, there's CD21. And the last piece of the co-receptor is CD81, which actually passes through the membrane. So here are the key players of that co-receptor complex. And I'm mostly going to talk about the role of CD21 and Ig alpha and Ig beta in B cell signaling. So CD21 and Ig alpha and Ig beta. So we haven't talked about it much yet, but we will talk about the role in complement and how complement tags pathogens. But for now, I'm going to say that complement, right, is going to tag proteins with this little bit of peptide, right? It's going to tag actually pathogens, okay, with that peptide, and that little piece of peptide, we're up here in the label now, apologize, is called C3D. Okay, and that's recognized by CD21. And this ensures that only a pathogenic antigen or non-self is going to be recognized by both CD21 and the B cell receptor. Okay, so this is an important kind of regulatory aspect. This also increases the avidity of the B cell receptor because we have multiple sites that bind this antigen. Okay. So we have an increase in avidity. So when all this happens, right, so we have signal reception, we see that CD21 is binding and CD19 and CD81 are also involved in this process, then we're going to get signaling inside the cell, okay? And where that signaling takes place is within the cytoplasmic tail of Ig alpha and Ig beta. So within the tail of these proteins are ITAM motifs, or immunotyrosine activation motifs, ITAMs, okay? And these ITAMs have tyrosine sites that are commonly or amenable to phosphorylation. So they're often represented as P for phosphorylation and Y for tyrosine, okay? So these are important sites in these molecules, okay? So kind of the take-home points here, again, is this cooperation between adaptive immunity and innate immunity, and it increases multivalency. And then you have the ITAM motifs within Ig alpha and Ig beta. Okay, let's now talk about the T cell receptor. Scooch this over a bit. Make sure I'm in frame. Okay, so here we have our T cell receptor. And this is composed of an alpha and beta chain. And the T cell receptor recognizes peptide presented by antigen presenting cells. So here we have our peptide, okay? And again, this alone doesn't cause signaling, but instead we have this co-receptor complex that's going to propagate the signal within the cell. So we have a couple key players here. We have CD3, which is actually 1, 2, 3, 4. Right, we have these 6 proteins that make up CD3. And I guess I can put these in color. And I'm drawing it on this side of the membrane, but it's actually surrounding the T cell receptor. And then we have the co-receptor CD4, CD8, depending on, what MHC molecule we have. Okay, so again, we have something very similar happening with the CD4 and the CD8, increasing avidity of this interaction because it's interacting with MHC. So that's an important component of this reaction. And within the cytoplasmic tails of CD3 are lots of ITAMs. So when this signaling pathway starts, right, these tyrosine sites are going to be phosphorylated, and once they're phosphorylated, okay, their role is that they serve as docking sites for other proteins. And the proteins that bind tend to have these domains called SH2 domains or PTB domains. And we'll talk more about this next stage in the next course, or in the next class, okay? So here we have, these are the domains that like these ITAM motifs when they're phosphorylated. Okay, so again, this doesn't just happen unless there's some sort of inflammation around, and that's because CD28, you also need this interaction between CD28 on the T cell. So here's CD28. And, you pick a color I haven't used, and CD80 or CD86 on the antigen presenting cell. Okay, and this is going to start this signaling pathway, okay? So to kind of, again, highlight this, in both cases, right, we have a co-receptor, right, that's increasing avidity. And again, in both cases, we have accessory proteins with these ITAM motifs that are going to get phosphorylated. The tyrosines in those motifs are going to get phosphorylated, and they're going to serve as docking sites for proteins that are going to carry on this signal transduction pathway. And I'll explain that in more detail in the next class. So that's it for the podcast today, and I look forward to seeing everyone in class. Thanks, bye.